Hello guys, this is Dr. Sambodhi and today I will be discussing about structure of skin. First we will be discussing about layers of the skin. Then we will be discussing about cells of the skin. Then we have glands. Then we have nerves and receptor of the skin. Okay. Yes, so that is what the gist is. So firstly we will be discussing about layers of the skin. In layers of the skin there are three layers of the skin, namely epidermis, dermis and hypodermis. So firstly let's start with the epidermis. The epidermis, what is epidermis? It is consists of multiple cells. These cells are called as keratinocytes. Yes. Now in keratinocytes, these are composed of intermediate filament. Intermediate filament, the predominant is being keratin. Now there are 54 functional keratins. Yes, functional keratin. Of this 54 functional keratin, we have 37 epithelial keratins and 17 hair keratins. These are approximately 40 to 70 kilodalton. Okay, so now we have types of keratin. Firstly we have acidic keratin and then we have basic keratin. In basic keratin we have K1 to K8 and then acidic you have K9 to K20. Along with it, in basic keratin with K1 to K8 you have chromosome 12 and acidic you have chromosome 17. These are your keratins. Also you have intermediate filaments. Intermediate filaments, now there are 6 types of intermediate filaments and there are others unclassified. So 1, 2, 3, 4, 5 and 6. So one is in intermediate filaments, as we discuss here, the basic keratin which is K1 to K8, these are type 2, K1 to K8, and the acidic keratin which is K9 to K20, this is type 1, K9 to K20, type 1 type of intermediate filaments. Now type 3 is your vimentin, your desmin, then your glial fibrillary acidic protein and your peripherin. Then you have type 4 which have your NFL, MH, along with it you have alpha intermixin, then syncoilin and cinnamon. These are your type 4. Then type 5 may you have laminins, your laminins that are defective in certain conditions like epidermolysis bullosa. So type 5 you have laminin AC, laminin B1 and laminin B2. Type 6 you have nestin and others. Others may filensin and fecinin. Now these are your types of intermediate filaments. Now you have the layers of the epidermis as stratum corneum, lucidum, granulosum, spinosum, basal. You can remember this as college SB. Now firstly the corneum layer, which type of keratin it is have? K1 plus K10. Yes, 1 and 10 is present in your stratum corneum, which is the horny cell layer. Then you have your granulosum and spinosum layers. Granulosum and spinosum में there is K4 plus K13 in mucosa. Also, as the cells will get cornified, they will express K1 plus K10. You can say 4 is equals to 1 plus 3. So K4 plus K13 is in granulosum and spinosum, and in basal you have to increase one number from this. So K5 plus K14. Also 5 is equals to 1 plus 4. So you can remember it like that, like this. So stratum corneum में you have K1 plus K10, granulosum and spinosum you have K1 plus K10 with K4 plus K13, and in stratum basal you have K5 plus K14. Now these are your keratins. Now let's going to the layers. So firstly you have stratum corneum. Stratum corneum is the outermost layer of the epidermis. What will it do? Function. It will provide mechanical protection. Also it will prevent water loss, so it will be barrier to water loss. And it will also prevent entry of environmental substances, right? Like your bacteria, microbes, everything. It will also prevent entry to that. Now this is in stratum corneum. It consists of keratin, which is in filaggrin rich matrix. Now normally stratum corneum cells have no nuclei, which is absence of nuclei. If there is presence of nuclei, then it is called as parakeratosis. It can be seen in incompletely keratinized cells, parakeratosis, as well as certain conditions like psoriasis. Then stratum corneum में cells are arranged in a bricks and mortar model. So you know how the bricks and mortar are laid down, like this is the brick and in between you have the mortar. Now the bricks are your corneocytes, and this mortar which is there is your extracellular lipid matrix. Extracellular lipid matrix which is made from loricrin. This is your mortar. Then after stratum corneum you have stratum granulosum, which is the second layer, and as the name suggests, granulosum, granules, it will contain certain granules. Now stratum granulosum contains keratohyalin granules and lamellar granules. Keratohyalin granules your keratohyalin granules will contain profilagrin, and loricrin. Yes. Now these profilagrin and loricrin will further be involved in the formation of cornified cell envelope. So filagrin, what happens is firstly the profilagrin is converted to filagrin, right? Profilagrin is converted to monomeric filagrin subunits, which is a calcium dependent process. Now this filagrin is responsible for aggregation and disulfide bonding in cornified cells. Aggregation and disulfide bonding. In cornified cells. So it will act as a glue. Yes, it will act as a glue and it will keep the cornified cells bind together. And then after this filagrin, it is degraded into urocanic acid and pyrrolidone carboxylic acid. Now these two, urocanic acid and pyrrolidone carboxylic acid, these acts as NMF, which is the natural moisturizing factors. Yes, natural moisturizing factors. So these will act as NMF, natural moisturizing factors. So what they will do? They will hydrate the stratum corneum, along with they will block the UV radiation. This is filagrin. Then we have again transglutaminase. Transglutaminase also act as a glue. They help in cross-linking the cornified envelope. This is also again a calcium dependent process like profilagrin to filagrin conversion. Now you have to remember three types of transglutaminase, which is TG1, TG2 and TG3. Now this TG1, transglutaminase 1, this is a keratinocyte transglutaminase which is membrane associated, and TG1 defect will cause lamellar ichthyosis type 1, lamellar ichthyosis type 1. Okay, so TG1 will cause type 1 type of lamellar ichthyosis, and also it will cause non-bullous congenital ichthyosiform erythroderma. So non-bullous CIE. Lamellar ichthyosis type 1 is due to which? TGM1 gene. We will see ichthyosis detailed in the later sections. And non-bullous congenital ichthyosiform erythroderma, this is due to TGM1 plus allox 12B plus allox E3. This is your TG1. Now transglutaminase 2, it is present in fetal epidermis. Yes, in the fetal epidermis you will see transglutaminase 2. Also it will be seen in basal layer of adult epidermis. Then you have transglutaminase 3. Transglutaminase 3 is present in hair follicles and differentiated epidermal cells. Now transglutaminase 3, you know this is important as it is present in sublamina densa, and the defect in transglutaminase 3 cause a very important disease which is dermatitis herpetiformis. You can see anti-tissue transglutaminase antibody in dermatitis herpetiformis, right? Now what is the process of epidermal differentiation or keratinization? These are all triggered by calcium, which results in the formation of cornified envelope. So your cornified envelope, what happens in cornified envelope? Firstly, so there is cross-linking of envoplakin, periplakin along the cell membrane. Yes, this envoperiplakin, they will get cross-linked along the cell membrane. Also there will be loricrin cross-linking of the envelope, and there will be keratin and filaggrin cross-linking. These all will be there to support the cornified cells. Envoplakin, periplakin cross-link along cell membrane, loricrin cross-linking, and keratin and filaggrin cross-linking. Also lamellar granules. Lamellar granule will be formed and extrusion into the extracellular space. So lamellar granules are granules, they contain ceramide, so they come between the stratum corneum and stratum granulosum, and then they extrude their contents which are mainly ceramides. So this was your stratum granulosum. Now stratum spinosum, third layer. Corneum lucidum, stratum spinosum is your fourth layer. Yes, there is one stratum lucidum between stratum lucidum between stratum corneum and stratum granulosum. Lucidum is just present in your palms and soles. That is the layer. Now stratum spinosum, as the name suggests, it will contain spines. Now in stratum spinosum, the cells will have rounder nucleus, more flattened appearance, and they will contain lamellar granules or odd-line bodies. Lamellar granules are present in both stratum granulosum and stratum spinosum. So these will contain lamellar granules. Also stratum spinosum, as we discussed earlier, they were expressed K5 and K14, and as they will become keratinized, they will express K1 plus K10, which is corneum is expressed. Also there are certain hyperproliferative markers. Hyperproliferative markers are your K6, K16 and Ki67. These are your hyperproliferative markers. So in the hyperproliferative conditions like psoriasis, actinic keratosis and wound healing, these hyperproliferative markers will increase, right? Actinic keratosis and wound healing, where the skin is proliferating rapidly, these hyperproliferative markers will increase. And also this layer has desmosomes. Stratum spinosum contains desmosomes. Desmosomes are the intercellular connection between the cells which hold the cell together. Yes. So desmosomes, what are these? They are composed of transmembranous desmoglein desmocholine pairs. Now there are 4 types of desmosomes. So you have to just remember DSG1, which is present in the upper epidermis, mainly between stratum corneum and stratum granulosum, which is subcorneal, and DSG3 is present in the lower epidermis or in the basal epidermis, so suprabasal. So in the pemphigus groups of disorder, the antibodies which are against these antigen will get, you will see the pathology there. Like in pemphigus foliaceus, the antibodies are directed against desmoglein 1, so you will get a subcorneal split. And in pemphigus vulgaris, where the antibodies are directed against desmoglein 3, you will get a suprabasal split in desmosomes. Then you have certain gap junctions in stratum spinosum. Gap junctions are more abundant in stratum spinosum compared with stratum basal. Now these gap junctions, certain disorders are associated with it, like you have connexin 26. The disorders are Vohwinkel syndrome. Then you have KID syndrome, KID. KID is your keratitis, ichthyosis and deafness syndrome. So keratitis, ichthyosis and deafness. You have KID syndrome here. And then you have palmoplantar keratoderma with deafness, PPK with deafness. All are associated with connexin 26 defect. Then you have connexin 30. The defect associated is hydrotic ectodermal dysplasia. And then you have your connexin 30, 30.3 and 31, which is associated with erythrokeratoderma variabilis. Erythrokeratoderma variabilis. Now these we have discussed till about gap junctions and desmosomes, right? Now lamellar granules. Lamellar granules are what? Membrane bound intracellular structure, which are 0.2 to 0.3 micron in diameter, 0.2 to 0.3 micron. Now these are responsible for forming ceramide. Ceramides, what does they do? They form the major lipid. They are the major lipid which are responsible for barrier function of the skin. Lamellar granules, they are seen in upper stratum spinosum and stratum granulosum. So firstly they will be in spinosum and granulosum layer, then they will migrate upwards between the stratum corneum and stratum granulosum, and then lamellar granules will discharge their content here. So ceramides will be discharged between the stratum corneum and stratum granulosum. So it will be present here, so it establishes a barrier to water loss. And also ceramides along with filaggrin, they mediate stratum corneum adhesion. They also secrete other substances like glycoproteins, glycolipids, phospholipids, free sterols and glucosal ceramide. So that is why your dermatologist will suggest you to use a ceramide based moisturizer. Also there is a golden ratio in skin which is 3:1:1, that is ceramides is to sterols is to fatty acids. Glycoproteins, glycolipids, phospholipids, free sterols and glucosal ceramide. What did we see there? Carboxylic acid, pyrrolidone carboxylic acid and urocanic acid, which were acting as natural moisturizing factors for your skin, which were present in stratum granulosum. Filaggrin was giving, and these lamellar granules are giving your ceramides. Now then, there are certain disorders which you have to keep in mind with respect to lamellar granules. First is your Flagel's disease. These are also important MCQ which were in NEET. Flagel's disease, you can see decreased lamellar granules. Then Harlequin ichthyosis. In Harlequin ichthyosis, you can see mostly abnormal or absent lamellar granules. Harlequin ichthyosis is due to defect in ABCA12, ABCA12 gene at 2q34. Then you have X-linked ichthysis. In X-linked ichthysis, you will get missing steroid sulfatase in lamellar granules. Now this was stratum granulosum. We will move on to stratum basal. Stratum basal is also known as stratum germinativum. Yes, it is the lowermost layer of your epidermis. These are the basal cells, which are like columnar cells, single layer cells like this. Also, basal cell contains ornithine decarboxylase. Ornithine decarboxylase is a proliferative marker. You also saw proliferative marker in stratum spinosum, which were K6, K16 and Ki67. In stratum basal, you have ornithine decarboxylase, which is the proliferative marker. Now, these are stimulated by trauma, UV, epidermal growth factors, estrogen, then your beta agonist, and your tumor promoters. These are all the things which will stimulate your ornithine decarboxylase, which will lead to hyperproliferation, and it is inhibited by retinoids. Stratum basal will express K5, K14, and also it is composed of microfilaments. You know microfilaments, which are actin, myosin, and alpha actin. Now, as they are the microfilaments, they help in movement. So, they will help in upward movement of the cells as they differentiate. Now, stratum basal plus stratum spinosum, both together is called as Malpighian layer. Malpighian layer. M equal to B plus S. Yes. Now, here our epidermis ends. Now, between epidermis and dermis, you have your basement membrane zone or dermoepidermal junction. Dermoepidermal junction. Now, let's see if this is your stratum basal. In stratum basal, you have your keratin intermediate filaments, and then here are your hemidesmosomes like this. So firstly we will name the components of dermoepidermal junction or basement membrane zone. Yes, so first it is composed of keratin intermediate filament, then your hemidesmosome, this is your stratum basal, then you have your lamina lucida, then you have your lamina densa, then इसके नीचे you have a sublamina densa. These are the components of basement membrane zone. Yes. Now this is the basal keratinocyte, this is the hemidesmosomes. In the hemidesmosomes you have these two plaques. Yes, this is the inner plaque and this is the outer plaque. Now inner plaque, what will happen at the inner plaque? Inner plaque will attach your keratin intermediate filaments to your hemidesmosomes, right? So hemidesmosome is the adhesion complex that attach basal keratinocyte to extracellular matrix. Now as we discussed, this inner plaque will attach to the keratin intermediate filament and this outer plaque will attach to the anchoring filament. Now the inner plaque is composed of plectin, then your BP230, plectin and BP230, this you have to remember, and your outer plaque BP180. BP180 is collagen type 17. Yes. Now BP230 is also called as BPAG1 and BP180 is your BPAG2, which is at the outer plaque. So inner plaque will be 1 and outer plaque will be 2. This is your hemidesmosomes and inner plaque. Now in BPAG2, this one, the NC16A domain of BPAG2. This is the target of autoantibodies in bullous pemphigoid. In bullous pemphigoid, this is the target of autoantibodies in bullous pemphigoid. Yes. Now then you have lamina lucida. This is the lamina lucida. Now this is the electron lucent region, so that is why the name. Now it has anchoring filaments. In lamina lucida, let's guess this is the anchoring filament. Now anchoring filament, it consists of laminins. So like this. Anchoring filament. Now this anchoring filament will composed of laminin, again BP180 and nidogen and your integrins. Here major laminin is laminin 332 and integrin you have alpha 6 beta 4 integrin in lamina lucida. Now lamina densa. Lamina densa is again, as the name suggests, lamina, so it will also contain laminin. Now you have your type 4 collagen, nidogen and your heparin sulfate. Now the prototype of heparin sulfate is perlecan. So lamina lucida, it's like this, and lamina densa. So here what will happen, these are your anchoring fibrils and this is your collagen in sublamina densa. In sublamina densa, the anchoring fibrils will be, these are the anchoring fibrils which are composed of collagen type 7, collagen. This binds to type 1 and type 4 collagen, laminin 332 and microfibrils. So these loops over these. This is your type 1, type 3 collagen, interstitial collagen. This is not a good diagram, but